In this section, we examine how scientists make the first synthetic life called Synthia. In section 2, we discuss some examples of gene design or redesign. With Synthia, the research team chemically synthesized the whole genome of a bacterial species called Mycoplasma mycoides. They trimmed off the so-called junk DNA and included only half of the genome with long genes with specific functions. Their report in Science in 2010 showed that the morphology and phenotypes are similar to the wild-type bacterial cells. They used an oligonucleotide synthesizer to make many 1,000 base long DNA fragments and combined them into gene cassettes, making more assemblies 10,000 bases long in yeast added up to 100,000 base pair, BP, and eventually they make the synthetic genome in 1 million base pairs of a circular DNA with many specific markers and restriction enzyme sites. The whole process involved 2 chassis to produce DNA, E. coli and budding yeast. They added some bacterial DNA from a yeast artificial chromosome so that the vector could be inserted into both E. coli and yeast. The synthesized DNA fragments were tediously added bit by bit into the vector, and eventually the whole synthesized circular DNA was created and put back into the bacterium with its original DNA removed. So, are we creating life? Yes or no? The DNA was chemically synthesized, and we need to use bacteria and yeast to assemble the chemically synthesized DNA. This chemically synthesized DNA was really just a copy of the original wild-type DNA with a few new marker genes added. The invention was made by Craig Venter, who later wrote a book entitled Life at the Speed of Light, in which he claimed that we can now split up evolution on our own course by adding and redesigning new genes for human use. So the question of whether we can create new life forms comes back again. What else can we do? What have we done so far in redesigning genes using DNA that could be synthesized on a machine or amplified with PCR or in cells that we can manipulate? So far, we can join different pieces of DNA together by ligation or by cutting and pasting the genes we need. We have plasmid vectors and artificial chromosomes from yeast to carry the inserted DNA into the host. These host cells, or chassis, nickname, could be bacteria, yeast, cell lines, or even organisms like fish or mice. We can create synthetic gene cascades in different chassis with corrected or customized codons. We can create gene networks that can work as a cascade of gene circuits. An abstraction of hierarchy of gene assemblies of regulatory networks is shown here. We have different parts making up the devices. Cascade of gene devices can build up modules, and many different modules could develop into a new system. So what are these parts? We call these parts as bio bricks. just like playing toys with the synthesized DNA adding into parts, device, and system. An analogy of using electric circuits to building biological parts. The products are different genetically engineered machines. All biobricks have different biological functions, such as gene promoters, reporter genes that express different colors, sense chemicals, or transfer chemicals. Tens of thousands of them could be cataloged in a synthetic biology registry online. This slide shows the websites for the main pages of the registered biological parks or biobricks with various functions, such as gene promoters, ribosome binding sites, translation units, terminators, and plasmid vectors. Here is a bacterial gene with a gene promoter using RNA polymerase or RNAP to transcribe genes. The transcription of genes can be first activated and then depressed by using or adding a repressor. We can then enhance gene expression by enhancing the ribosome to bind for more efficient translation to produce protein. With these strategies in mind, we can redesign many different genes to fit any purposes. The question is, what would we like to make? Here is a scientific idea that could be interesting. It may not have immediate use, but in this case, E. coli was engineered to sense light, and a membrane photoreceptor was added to the membrane to sense red light. The sensor protein was added artificially with an intracellular histidine kinase domain to activate the expression of a repressor on the promoter C or POM-C promoter linked to the LAC-C gene, resulting in a black output. In the engineered bacterial plate, the researcher used a negative film and flashed it with light, which repressed the gene expression to give an image without the LAC-C. Without light, the LAC-C enzyme can produce a black compound, forming the black and white film image on the agar plate. A similar circuit can also be made in mammalian cells. In this circuit, a viral gene promoter of SV40 is used to drive the expression of several proteins, including VP16 and other proteins. However, SEAP, which is an alkaline phosphatase, is under the control of a different promoter that could be activated by VP16 by adding a photorectin repressor. Therefore, photorectin can act as a repressor of SEAP. The second example is the use of urate transporter to bring in urate and SV40 to express a repressor controlling the expression of urate oxidase. If urate gets inside the cell, urate oxidase is produced, and over time, urate can be degraded by the oxidase. The last example here is making use of auxin to degrade proteins and using an auxin receptor linked to SCF with ubiquitination and the auxin transcription repressor 8, which also binds auxin. If the repressor is linked to a target protein, the addition of auxin will cause the protein to be linked by auxin with a complex, and the target protein could be ubiquitinated for further degradation. After reading about these ideas, you can design your own genes with biobricks. Here, synthetic biology involves the use of standard parts to perform genetic engineering. Biobricks mimic biological components like electronic parts that can easily be assembled. The iGEM competition from MIT began as a course and has now become an international competition with more than 300 teams from around the world taking part. You can check out the iGEM teams and their ideas posted on their wiki pages. Today, the iGEM competition has over 10 tracks, covering medical diagnosis, energy and environment, food and nutrition, information processing, etc. If you are interested to start a team, please go to their website. But first, you need to brainstorm your project ideas, your team's project idea, later making your biobricks and doing the experiments, and make your presentations in Boston. However, before you start, think about how will the general public interact with your project, or how can you design for a project to solve global problems. Let's watch the last video, section 4, on synthetic biology and our society.